Hello, hello,
Hello?
hello.
Hello?
Hello?
Am I audible, sir?
Am I audible, sir?
Hello, Akhil.
Yes, sir. Yes.
You can now it is audible. You can start.
Okay. Thank you. Thank you, sir.
Respect.
Respected Chairman and all other invited dignitaries who are here, a warm good morning to all. My topic of research is reproductive dynamics and conservation of Humboldtia brunonis, an endemic tree species of the Western Ghats.
Western Ghats
Western
is
Ghats
one of the
is one of the major. It
includes
includes host of unique flora and fauna, and including several endemic species. And Humboldtia brunonis is one of them. And Humboldtia brunonis, despite having its reproductive biology, uh, no other more studies which are carried out in the field of reproductive biology. So, the particular study mainly focuses on its reproductive dynamics, and it is very much important to predict its survival and fitness in its natural habitat. Introduction, in which in Cambodia it includes 8 species and 2 varieties, in among which 7 species and 2 varieties are endemic to India. The remaining one species, which is Humboldtia lorifolia, is endemic to Sri Lanka. And here in 6 species and 1 variety, which grow below the Palagar gap, here the selected species Humboldtia brunonis variety brunonis and Humboldtia brunonis variety rectapushpa population was found above the Palagar gap, and it is considered as a semi-myrmecophyte plant. Usually, the myrmecophyte plant, which means the plant have such a strong interaction with ants. Here, the ant species, several ant species are present in the particular plant. It is considered as a semi-myrmecophyte plant because uh, the reason I will explain later. These are the major objectives of the study. Penology and floral biology with a special reference to flowering and fruiting. Pollination mechanism and reproductive success. Identification of causal factors for endemism. To understand the breeding system. To study the seed biology and germination capacity of the plant. Molecular analysis, propagation and conservation. These are all the titles which I was covered as part of my review collection. Uh, this particular figure shows the distribution of both the species. Humboldtia brunonis variety brunonis was distributed in Tamarashiri, Kakkayam, Aralam, Brahmagiri, Unikal, and Agumbe forest areas. But the special variety Rectapushpa was strictly endemic to Kakkayam forest areas. Several field trips were conducted and located the populations of candidate species in the forest areas of Tamarashiri and Kakkayam of Calicut district. Phenology, the flowering pheno events, the following pheno events were recorded according to Daphne et al., such as flower initiation and duration of development, peak flowering period, the time of flower opening, that is anthesis, was observed, flower longevity, fruit initiation and development, fruit maturation and dehiscence were also observed. Then the soil properties of both the selected areas were carried out, and floral morphology, which includes floral or flower order detection and flower volatiles, SEM analysis, quantification of pollen ovule ratio based on crude oil, and pollen fertility and viability was analyzed with the help of different staining techniques such as and Alexander testing and fluting. Pollen germination studies were carried out both in vivo and in vitro. In vitro pollen germination studies are carried out under Brubaker and quarks medium with a different concentration of spores. Stigma receptivity was also analyzed by H2O2 test and BAP test. Then pollination studies are carried out in which anthesis was noticed and recorded. And then the activity of the pollinators were observed. Plant animal interactions were studied. Breeding system, such as open pollination, autogamy, heterogamy, and xenogamy experiments were conducted. Fruit and seed biology, which includes fruit seed ratio, seed moisture content, conductivity test, and germination studies were carried out. Molecular analysis was carried out with the help of ISSR markers, and clonal propagation was done by using the rooting hormones such as IBA and NAA. Results. Humboldt brunonis variety brunonis. The selected population of Humboldt brunonis variety brunonis located on Tamarashiri on either side of the road. Most of the population are distributed in the Tamarashiri. And the particular plant species which is endemic to Kerala and Karnataka. These Brunonis, variety Brunonis is a moderate tree. The leaves are four foliate. The leaves are pubescent when young, and the stipules are present. Inflorescence are raceme. Flowers white, pink, pale pink in color. Sepals are five. Petals three or five in number. Staminodes are five. Ovary pubescent. Fruit spores, and it contains seeds, which is one, two, or sometimes three. Distribution in Kerala, which is distributed in Kerala, especially Wayanad, Tamarashiri, Kakkayam, and Kannur. Karnataka, or this particular species was located in Hulikal and Agumbe forest areas. This is the habitat of variety Recta Pushpa. Humboldt brunonis variety Recta Pushpa. Comparatively, shows a shrub-like nature when compared to the brunonis habit. And here in the figure, we can see in this particular size, the flower arises. That means this particular plant is reproductively mature, even in this small size. And this particular species is strictly endemic to the Kakkayam forest areas. These are all moderate trees on to small shrubs. Here the leaves are four foliate, pubescent when young, stipules are present, inflorescence and raceme. Flowers are red in color. The sepals five, petals three, stamens five, staminodes are also present, which are five in number, and the fruits which are cones, and it contains seeds which are one to two seeds are present. These are the major observations regarding the morphological difference between these two varieties of Humboldt brunonis, in which Humboldt brunonis variety brunonis is having a height up to 6 to 14 meter when compared to variety brunonis recta pushpa is a small tree. It is only up to 4 to 6 meters in height. In variety brunonis, they produces the inflorescence which are 10 to 20 centimeters in long, and in recta pushpa, those inflorescences are also short inflorescence. And in the flowers, Humboldt brunonis variety brunonis produces white, pink, or white tinged with the pink flowers, and in recta pushpa, they produces only the red flowers. Here, in which both they produces the stamens, and it contains this. Also, the staminodes are present, but in variety brunonis, the staminodes are filiform, and in variety recta pushpa, they are globose in nature. Then the is long in bodies and which is short in variety recta pushpa. In the petals are broadly obovate in variety brunonis and which are narrowly obovate in variety recta pushpa. And in Humboldt brunonis variety brunonis, it is having 1 to 3 seeds, and in variety recta pushpa, they produces only 1 to 2 seeds. The soil properties of both the selected were carried out, that is Tamarashiri and Kakkayam, and both the soil samples are slightly acidic. They are non-saline with a high amount of percentage in organic carbon, and the available phosphorus, available potassium is also similar, which is medium, but the available phosphorus content is varies, which is higher in Kakkayam when compared to the Tamarashiri sample. And here also the iron content. Iron content also varies in Kakkayam. In Kakkayam forest areas, the soil contains more when compared to Tamarsheri. And the one, both the soil samples are having plenty amount of water holding capacity.
These are all the associate species of the
Associates
selected
of
particular.
particular Hamburger Pronotis variety Pronotis and variety
Here in which
Pushpa.
Gonium thalamus,
Here in which Gonium thalamus vinadensis, Cinnamomum macrocarpum, and Xanthophylla are known here. These three species are only associated with the Hamburger Pronotis variety Pronotis. That means they are located at Tamarsheri. And the other species from Bellophora frondosa and so on, Shythium brutum, all species are the associated species of Hamburger Pushpa located here. And the Totia species, Totia Shivaraj and Totia Shashidharaniyan. Both these Totia species are present in both Tamarsheri and Kakayam. Floral variation was observed in the variety Brunonis. Means the petal color, it may be pink, white, pale pink, and white with a pink tinge on the base was observed. The sepal color also changes. It may in some flowers they have pink sepals, and some of them are pale pink, and some are pale green and red in color. The petal number also varies. Petal number maybe 3 or 5. Usually, most population of Hammonis variety Brunonis and Tamarsheri having 3 petal flower. The 5 petal flower that was only located at Aralam. The of the internode may be sometimes compact or loose. Whether it is compact, then the size of the inflorescence is shorter. Whether it is loose, the size of the inflorescence is larger. Then the flower per inflorescence also varies. If the inflorescence is higher, longer, then it is more flowers in it. That means less than 20 flowers, or some inflorescence contains more than 30 flowers in a single inflorescence. The stamen color also varies. Some of them is red and some are yellowish pink and pink in color. This type of color variation common among the other species of Hamburger also. Flower and fruit quantification. Total number of inflorescence per tree in Hamburger Pronoris is comparatively a larger tree and produces more branches when compared to variety Rectopushpa. So it bears more inflorescence per tree, which is approximately 250 plus or minus 75. But in Rectopushpa, they contains only 35 plus or minus 7 flowers per tree. Number of flowers per inflorescence may also varies, and total number of flowers per tree has a significant ratio here. We can see Hamburger Pronotis produces more flowers per tree, while in Rectopushpa, comparatively it is lower in number. The number of pods per tree is also higher in Pronotis and lower in Rectopushpa. Then the percentage of fruit set and the viable seed percentage was also very fewer. The percentage of seed germination was very much lower in both the species. Phenological events such as leaf initiation, leaf maturation, bud maturation, anthysis, fruit initiation, fruit maturation, fruit dehiscence were observed. This is the graph which shows the phenological calendar of the selected species, both the varieties, Hamburger Pronotis and variety. The plant usually starts flowering in October and extends up to December and reaches peak during November. Pushpa starts from the middle of October to December. It reaches a peak in the last of November. Here in the graph, the last three, yellow, purple and red, which shows Hamburger Pronotis variety Pronotis from Tamarsheri and Kakayam and Rectopushpa. These are actually the selected area for the study. And the other two are the same plant which was located in different location, which is Aralam and Ulken, but the flowering season with respect to the place of occurrence. Then The flower shifting was also noticed in the selected species. The study was carried out under 2018 to 2023. And during 2018, Humboldt Brunonis here, the green shows it starts in the month of August, and after continuous observation, there was a notable flower shift, and the flowering pattern shifted to the next month. So the inner yellow and violet changes starts flowering from the next month, and the outer three rings show the flowering color of during 2018 and 2021, they start flowering in a particular month, and that particular time was shifted to the next month, or the outer circle. That's why the red circle was changed from here to here. Flower shift in its time of flowering. This is the slide. It shows the flower development from initiation to maturation. Here, the flower took 30 days for full opening. Both the varieties took 30 days for full anthesis. Analysis was carried out with the help of GCMS, and these are the major floral volatiles which are observed in the GCMS analysis, among which beta is the major olfactory produced by both the species, both the varieties of Humboldt Brunonis, in which beta osmine content is higher in variety Brunonis when compared to Rectopushpa, but the sulfur containing compound dimethyl sulfide is higher in Rectopushpa compared to variety Brunonis. This olfactory compound beta osmine is a plant-derived sesquiterpene, and it will mimic the pheromone produced by the honeybees. Based on the literature, it is clear. And in this picture, we can see some honeybee species which are visible in flora. And the right side, we can see the GCMS graph. Beta osmine content which is present in Humboldt Brunonis variety Brunonis, and here this graph shows the TCMS analysis of Humboldt Brunonis variety Rectopushpa. Here the dimethyl sulfide, a compound that is morphology, pollen dehiscence, viability, fertility. Pollen, then ovule was isolated, and it is anatropous in type, and pollen ovule ratio was calculated, and stigma receptivity were also observed. Pollen biology includes pollen morphology. The pollen grains are tricolpate with elongated furrows on the surface, and there are is 33.73 to 35.69 micrometer. Pollen grains are ellipsoidal pores and spherical to subprolate in equatorial view and angular in polar view. The exane thickness is 3.45 micrometer, and both the pollen grains have rugulate ornamentation on the surface. Both the varieties of Humboldt Brunonis, the pollen grains does not show any significant variation. Estimation of number of ovules per flower. Only two and three ovules are Humboldtia, and in which 6% of Humboldt Brunonis variety Brunonis have only one ovule, and most of the Humboldt Brunonis plant, that is 92% of Humboldt Brunonis, have two ovules, and two of them percentage of Humboldt Brunonis have three ovules. But in Rectopushpa, they also of the Rectopushpa plant produces two ovules within the flower. The three ovules which are not observed in variety Rectopushpa. Then pollen ovule ratio. The number of pollen per anther and pollen flower per flower. Pollen ovule ratio. There is not much variation or significance in between these two species, and Then pollen fertility analysis was carried out. Pollen fertility analysis was carried out with the help of two different strains such as acetocarbon and glycerin. Then Alexander's strain. Most pollen grains are on the day of anthysis at the time of flower opening, and the pollen fertility decreases the day after anthysis. Here, the above figure shows acetocarbon glycerin test. Pollen grains are fertile, they absorb more stain and it will more visible as red in color. If it is non-fertile, it will not absorb pollen grains stained in the figure. By using Alexander stain, if the pollen grains are fertile, they absorb the Alexander stain and the color changed into a purple. If it is not fertile, then the color will be a pale green in color. So we can calculate the pollen fertility. Then pollen viability. Pollen viability also analyzed with the help of FCR and DAP test. Here, just like fertility, the viability, most of the pollen grains are viable on the day of anthysis, and the viability of the pollen grains excessively reduces on the day before anthysis. And here, the above figure it shows the pollen grains which will more absorb the stain, especially FCR, and they produces a fluorescence. That pollen grains are considered as viable. And in if the viable pollen grain absorb the DAP stain, and they may produce a rust brown color. If there is no color, that means the pollen grains are not viable. If the pollen grain produces color, grains are viable. Then comparative analysis of these pollen viability and fertility compared with the in vitro pollen germination, from which acetocarbon Alexander strain is more significant when compared to other strain with respect to the in vitro pollen germination value, and DAP test is not much reliable for this. From this comparison, we can clearly say that DAP test is not much reliable, but acetocarbon Alexander strain is give more precise result when compared to in vitro pollen germination. Stigma receptivity was also observed. Stigma receptivity was also observed in bud stage at 2 days before anthysis and on the day of anthysis with the help of hydrogen peroxide. We apply the hydrogen peroxide on the stigmatic surface. If is receptive, it produces bubbles. On the amount of bubble emerges from the stigma, we can calculate which is receptive, how much receptive. In this sense, we apply hydrogen peroxide on the bud stage. Here they have a weak positive response that indicates a one positive sign here. And on the successive days up to on the day of the stigmatic surface that means they are very strong positive response to hydrogen peroxide, which means the stigma is more receptive on the day of based on the bubbles. And in vivo pollen germination studies are carried out in the field. After those pollination process, we collect the stigma and which was here in DAP and lactophenol cotton blue were utilized to stain the stigmatic surface and the grains and then observe under microscope. And regarding the result, on the day of anthysis, most of the pollen grains are produces more stigmatic and germination of capability of pollen grains will successively reduce on the day of anthysis. After the day of anthysis. Then pollen germination. Pollen germination studies was carried out in grow by mass medium containing different concentration of sucrose, in which 10% of sucrose is more feasible for the most of the pollen grains germination in both species. And here we can see the pollen tube where emerges, and these pictures are shown in taken from the 10% sucrose medium, and they produces the pollen tube here. And on the right side, It is also contains the in vivo pollen germination figures. Pollen grains are adhered on the surface of the stigma, and they produces the pollen tube over there. And those figures are actually for in vivo pollen germination, and the left side it is for in vitro pollen germination studies. Then pollination biology. Both the varieties of Humboldtia brunoris are mass blooming, and the flower opening time was noticed in the morning 4 a.m. to 6 a.m. And the adolescence was happening after the full blooming, that means 6 a.m. to 9 a.m. Then the visitation time, floral visitors are coming to the flower from the morning 6 a.m. to the next day, up to the next day evening. And the floral visitors are attracted towards the flowers because of the cue, that is the visual signals, and the plant also produces brood eggs for several organisms. And the flower produces nectar. The nectar secretory cells are present in between the calyx and the ovary. Mass blooming and nectar guides are the major advertisement by the flowers for the insects. Then, when studying pollination biology, these both the varieties of Humboldtia have a landing petal, and which are shown in the figure, landing petal, and which is not interrupted by the stamens, and most of the landing petal are all the petals are pink the base, but the landing petal has a broad line of marking. And the white petal, if the petals are white, then the sepals are more attractive, and which are pink in color, which will attract the pollinators towards the flower. And these are the floral diagram, which shows the landing petal on the top here, and the green arrow mark, which shows the presence of anther. In this figure, we can see the landing petal, which is not interrupted by any stem. So it's more visible, and the pollinators can easily land on the petal. This is sequential image of anthrodehiscence. Anthrodehiscence was starts from 6 a.m. and properly dehisces longitudinally, and it may lead up to 9 a.m. And beginning it is a slow process that anthrodehisces longitudinally, and then after this after 8 a.m. up to 9 a.m. it is a time in between 10 minutes, it is a fast process, and they're completely dehisce and pollen out. Major floral visitors: honeybees, Gonula species, Bombicetra humboticola, and butterflies are the major floral visitors. The flower elicits certain chemical substances which will attract insects and at the time of anthysis. Pollen, nectar, visual signals, and brooding sites constitute the primary reward for the visitors. All the floral visitors except ants foraging however, the ant species such as Bombicetus humboticola, Technomermicus albibus, Aniplolepsis graslips patrolling 24 hours in the flowering branch of the candidate species. The selected species, Humboldtia brunonis, variety brunonis, and variety rectopushpa, both the plant has a special structure, have a special structure called a domatia. Domatia is usually found on the leaves of some plants, but here the domatia is seen on the internodal region. Hence, it is called the colline domatia. It is a stem modification, and it is the site for mutualistic relationship with several organisms of the plant. For example, Bombicetus humboticola, Technomermicus albibus, and Aniplolepsis. These are all the ant species which are inhabited in the domatia. Plant provide brooding site for this ants. And there is also another one, arboretum, which is known as petioles, which is also present in this domatia. This domatia is actually act as the shelter for ant and arboreal earthworm. Sometimes these two organisms are present in a single domatia, but at that time there is a partition. We can see their brooding sites, and in the form of a septa. then the ants will protect the plant from herbivory, and in this interaction, both plants and ants are benefiting. The number of domatia may vary. It may be 20 to 30 domatia was observed in Humboldtia brodonis variety brodonis, and 5 to 10 domatia are present in Rectapushpa because that plant is comparatively smaller. Uh, Another one important feature which is extra floral nectary. Extra floral nectary, which means the nectaries which are present other than the floral nectary. Usually these extra floral nectaries are present on the lower surface of the leaves and also present in stipules, bracteoles, calyx, and they produce a sugary substance. And those sugary substances will attract all these insects, especially the ant species, during the off season. The ant is always present in the plant, even the plant produces flower or the flowers are absent. The ant will be there in the plant because of this particular structure. It will gives nourishment to the ant which are inhabit the plant species. And here the last picture it shows the seminalis, image of external nectary. It is a disc-like structure present on the lower surface of the leaves. Then, in both the varieties, extra floral nectaries are present on the base of the appendages. It is 2 to 4 in number, and it is usually present on the peripheral part of the leaves. The number of extra floral nectaries may vary. It is 8 to 14 in variety Brononis, but in variety Rectapushpa, it is 6 to 8 in number. Then extra floral nectaries plays a very much important role in particular ant plant interaction. For example, this ant species such as Opiscetus humboidicola and other two species are always present in the plant and everywhere in the bud, in the flowers, in the younger pods, leaves, everywhere the ant species are present. Because the extra floral nectaries are present bracteole, inflorescence, and stipules, etc. And they produces the sugary substance. And while that will patrolling these plants, and here in this figure we can see that visited the near to the extra floral nectary where does the sugary substance were produced. And in this picture, the ants are present on the fruit, and there are also the pests are also present on the fruits. Here in the right side, we can see the action of ant interaction. That is a white insect pest which is called uh, Planococcus citri. It is a pest which is common in Italy, and that particular pest is present on the younger pod, younger fruit of that particular plant. Then the ant species Technomyrmicus albicus is there, and it will disturbing the pest, and it will take away the pest from that particular young developing seed. Then some other interaction neither benefits and nor harm the plant. Then such as the of extra floral nectaries. They visit the plant only for extra floral nectary or the sugary substance provided by the plant. And the plant also produces shelter and for non-pollinators. The plant has extra floral nectaries on leaves. It produces a sugary substance. It attracts the bees, wasps, and other butterflies to the plant. The ants are primarily benefited by obtaining the sugary syrups and leaves. In return, they give protection from the herbivory and the pest also. Some other organism may cause damages to the plant, such as leaf damage, flower damage, and fruit damage. Uh, um, and sometimes they are not pollinators, but they will affect the plant's fitness, such as some pests were observed in the inflorescence of Rectapushpa, and some caterpillars are these are caterpillars which are identified, and some snail species which are also ate the leaves of Humboldtia. And leaf predation is in selected species. The caterpillars that will degrade the younger leaves and the mature leaves also. And some lichen species was also found on the leaf surface of Humboldt, which is identified as Stridula species. And presence of this particular lichen may affect the photosynthetic capacity of the plant also. And fruit predation is very much common in the selected species. For example, here the bonnet macaca or macaca radiata, which will consume the younger pods or the immature fruits from its natural habitat. It really affect the population, and some of them are also infected by the caterpillars, and they will not produces the viable seeds because of this particular infection. Malabar giant squirrel also consume the fruits of Ambrosia in its natural habitat. Nectar analysis was carried out for the selected species. Here in the figure we can see the droplet which are present in the figure, and which are produced in the nectaries. The nectaries are located below the ovary, and they produces the floral nectar. And these are the characters such as the position of nectary which is below the ovary, and when compared to the nectar of Humboldt, Humboldt Brunner's variety Rectopushpa produces more energetic value of the nectar compared to Brunner's. And the major components of nectar, such as sucrose, is little bit higher in Humboldt Rectopushpa. Then amino acids like the proline and alanine is more in Humboldt Brunner's variety Brunner's when compared to Rectopushpa. Then breeding experiments, open pollination, autogamy, heterogamy, xenogamy, and apomixis was carried out, among which xenogamic pollination gives better result for this particular plant, and that means this plant favor more cross pollination for its natural survival. And these are the figures shows while from the field observation during the field breeding experiment. The first one shows the insect trap, and the next one is the flowers are covered for breeding experiment. And in open pollination, most of the flowers are pollinated by the insects, but finally the rats will remain. There is no sign of fruit initiation or fruit production. Even though the plant produces more number of flowers per tree, but they produces very much lower fruit set in its natural habitat. Breeding experiment in Hamboldt Rectopushpa also favors xenogamy or cross pollination rather than any other pollination mechanism. And here the picture shows the breeding experiments which is conducted in the field at Kakayam. And here in the figure, we apply a gum on the branch, which will prevent the entry of ants into the inflorescence. Because ants are very much common in this particular plant. So whether I need to check out whether it is a pollinator or not. So we apply a gum on the branch which restrict the entry of ant and fluorescence with a polythene bag. When the flower opens at the time of anthysis, the polythene bags removes and the natural pollinators will attract it towards the flower, and then process of pollination was done. Then the flower again covered, and then some of the flowers produces seeds. And then fruit and seed biology. The fruit is a legume or pod, which is most common in both species, which are green in color. The size and the shape of the fruit is almost similar. Then the fruit viability was analyzed with the tetrasodium staining test, and most of the seeds are viable. Fruit seed biology. In the candidate species, the flower-fruit ratio is comparatively low. Most of the conditioned fruits does not have viable seeds. That is, the ovules may be aborted, and hence the pods does not contain viable seeds. Then the seed biology. The moisture content of the seed is higher at the day of harvesting. It is 41.5 percentage. It will successively reducing after harvesting. On 24th day, it is finally 20.4 percentage. And in connection with the electrolytic leakage activity, when the water content reduces, it may lead to increase. There is an increase, you can see electrolytic leakage affect the germination percentage. Most of the seeds are 96.2% of germination was in the freshly harvested seeds. The days passing, the percentage of germination will also decrease. So here in Rektapushpa also, most of the seeds are contains more water on the day of harvesting, and electrolytic leakage increases on successive day after harvesting, and it will affect the germination. This is the figure. It shows genetic variation of the selected species. Here, the H1 is Rectapushpa, and H2 is reproductively mature Brunonis, and H3 is a which is located near to the parental progeny. And there are following ISSR markers such as 854, 820, 864, 901, 857, 810, and 870 are used. Seven ISSR markers were used, in which we can see the H2 and H3 bands similar when compared to the other. For example, here H2 and H3, they have a similar type of bands because they are having similar kind of DNA within it. When compared to H1, which is Rectapushpa, and here these bands are absent in H1, and here in through which clear that all the progenies which are surrounded in the natural habitat are actually clones are which are similar when compared to the DNA analysis. Clonal propagation, that is rooting experiments with the help of rooting hormones such as NAA and IBA was carried out. Different concentration of NAA and IBA was carried out, and in which IBA concentration, especially 500 parts per million concentration of IBA, gives more response. 95.83 percentage of such clones are survived in the habitat. Most then these are the figures that show various conservation propagation techniques that I was carried out. Air layering was carried out. There is they does not produce any and seedlings are propagated in the conservatory, and then the right figure it shows the rooting experiments with the different concentration of Na and IBA, among which IBA 500 ppm gives better result, and they produces roots on the vegetative parts of the cutting. These are the major threats, such as developmental activities such as construction of development which is happening in the road. Human interventions may also disturb the population in its natural habitat. Natural calamities such as flood and non-seasonal rainfall also affect the population in its natural habitat. Then, for the conservation of this particular endemic plant, in situ conservation was carried out in our nursery JND Vidyalaya, and saplings were also distributed, also in the exit to in its natural habitat, and awareness were also carried out with the help of forest department, and these are the key findings. Five petals in Hambolcia Brunonis variety Brunonis was observed. color variation within Humboldt Brunneries, variety Brunneries was observed. Some of them are have flowers, some of them have pink flowers, and phenology vary in each locations. Two ovules are most common in both varieties. Ovules is anatropous. Beta osimine is a major compound in flowers. Both the varieties, the pollen morphology is similar. Pollen grains germinate in 10% solution in vitro. Floral visitors are less when comparing to the extrafloral nectaries. Honeybees, tetragonella species, Bombycetus fructicola, and butterflies are the most common visitors. Three type of plant animal interactions that are noticed. When the plant severs xenogamy, it needs an external agent for successful pollination. Most of the condition, the fruits does not have viable seeds. That is, the ovule may be aborted, and hence the pods does not contain viable seeds. And arboreal lives are together present in the Rhobacia. Leaf predation and fruit predation is high in Humboldt Brunneris variety Brunneris when compared to Rectuspa. Clonal propagation gives best result when the stem cutting treated with the 500 ppm of IBA solution. And activities is a threat for the species. In the present investigation, we observed that this species have very less longevity. Non-seasonal rainfall has a negative impact on the plant as they washes out all the full-blown flowers. Scarcity of pollinators and low insect visitation rate, that is, they are not sufficient to pollinate all the flowers in the population because most of the insects are visited the plant only for the extrafloral nectaring, and they took the sugary syrup from the extrafloral nectaring, and they will not perform pollination. The plant offers both food and brooding sites for the ants, and they provide protection from herbivores, hence they exhibit a mutualistic interaction. During fruit development stage, bonnet macaca and Malabar James squirrel eat the tender and the inflorescence adversely affect the population. Flower fruit ratio is very low. Only 1 to 3% of percent was noticed in the natural habitat. In most cases, seeds are not developed in pods. Molecular analysis showed that parent and progeny grown near to the root shown same gene flow, which confirm most of the progeny grows from the root of the parent. Progeny germinated from the seeds are less because of low seed production. Abiotic factors for poor distribution of habitat degradation due to natural calamities, construction of buildings, or expansion. All these factors, either alone or in combination with others, are responsible for the narrow distribution of the species in its natural habitat. These are the articles published and attended conference papers. I thank my research guide Dr. A.K. Srigala for giving me a support and guidance. I also thankful to the directors of the TGRI for providing facilities to carry out my work in time.
Agil,
Oh, sir.
you can give vote of thanks interaction.
Okay, sir. Okay.
So now the session is open for discussion or any suggestion or clarification from Mr. Agil. Audience can interact with Ahil for any suggestion or clarification from this work.
Congratulations, Akhil. You have done it very well.
Thank you.
First of all, I congratulate you have made a good work. The nature observation you have made of window in the very good study. Just I want to know that you have reported floral color variation.
Yes, sir.
Color.
Yes, sir.
And regarding visitors for floral interaction with other organisms, I will find any variation with floral visitors and floral color.
With respect to
Is there any selective visiting with particular floral color?
Sir, in both the varieties, Humboldt brown is variety brown and variety Rektapushpa. Honeybees and butterflies are the major visitors, and they also serve pollination. But the color variation is not significantly affect the visitation of the pollinators. There are some other factors also contribute to attract the pollinators, such as the